Okay, so this is epigenetics part 2. Again, epigenetics is the study of how we turn our genes on and off. Not necessarily the genetic code itself, but how we turn genes on and off. So one of the major ways that we turn our genes off is through a process called methylation. Now, this is adding a methyl group to DNA, and when we add methyl groups to DNA, we shut the gene off. Now, one of the ways that we produce these methyl groups is through this molecule SAMe. Now, when we convert SAMe to SAH, we produce a methyl group. Now, to make SAMe, we start with methionine. Now, methionine has a pink box around it to indicate that it is an essential amino acid. So everything in pink here is something we have to get from our diet. So we start with this essential amino acid methionine, we add ATP and magnesium, and then we can get SAMe, this molecule that produces these methyl groups for us. Now, when again it produces a methyl group, it's converted to SAH, and from there, SAH can be converted back to homocysteine, where it can then be back converted to methionine. So this is called the SAM cycle. Now, to convert homocysteine back to methionine, it can either be converted by trimethylglycine, and in the process it produces dimethylglycine, or it can be converted back to methionine with the help of B-12 folate in this enzyme 5-MeTHF. So we have two routes where homocysteine can be back converted to methionine, completing the SAM cycle. Now, however, homocysteine can then be can be also converted to cysteine, which can then be converted to glutathione and taurine. Now, glutathione is an antioxidant, and taurine is used in the liver to produce bile salts, and it also has antioxidant properties. Now, when we suffer from high oxidative stress, it is likely to drive this homocysteine toward this, the creation of cysteine, so we can create these antioxidants to clear the free radical buildup from oxidative stress. So if you are suffering from high oxidative stress, you are also suffering from undermethylation, or very likely to be suffering from a condition of undermethylation. Now, cancer proliferation is dependent on low methylation levels. So now you can see a connection between cancer and oxidative stress, because again, cancer proliferation is critical with low methylation levels. Now, some of the big environmental oxidative stressors that we have to face every day is toxic metals, pesticides, cigarettes, radiation, industrial waste, and smog. So you can actually now see a direct link, potential link here between these oxidative stressors and cancer. Again, because these oxidative stressors are likely to drive the homocysteine towards cysteine, away from the SAM cycle and away from us producing these critical methyl groups to shut our genes off correctly. Now, one way that we can remediate these oxidative stressors in these free radical buildups is through antioxidants. Now, we have two antioxidant enzymes, superoxide dismutase and catalase. So superoxide dismutase actually exists in two forms in our body. So we have superoxide dismutase that exists in the eukaryotic cell itself, which is basically our mammalian cells, and that requires zinc and copper. Now, we also have a superoxide dismutase that occurs in the mitochondria, and that requires manganese. Now, again, these are in pink, so you'll have to be getting these from your environment. And again, superoxide dismutase is an enzyme which is made of protein. The other enzyme, again a protein, is catalase. Now, catalase has a heme group, which is a prosthetic group, and this heme group that allows this catalase to fold and function properly. And this heme group, again, requires an iron molecule or atom. A couple of other antioxidants I have on the board here are selenium, vitamin C, vitamin E, even melatonin has antioxidant properties in the brain. glutathione, obviously, and then CoQ10. So here are just a couple of the antioxidants outside of these two enzymes here on the board that I have as options to help reduce oxidative stress. Now the takeaway here is, again, high oxidative stress is likely to lead to undermethylation, which prevents our genes from shutting off when they are supposed to. And it's, again, these low methylation environments that are essential for cancer proliferation.